There was a debate about whether the immune system care about cancer. But I think recent developments in the field have answered this question loud and clear. That is, cancer develop partly because of failure of the immune system. So as a result, there's a tremendous excitement and promise that focusing on rejuvenating the immune system will be one day the curable strategy for eradicating cancer.
When I started training in cancer immunotherapy, I had to walk down this long hallway of patients that were getting treated with adoptive immunotherapy. And Steven Rosenberg would update us on what's going on with these patients, and which was invigorating because back then, long ago, we would see large tumors regress and never come back. So it's not just now. It's been an exciting thing that I feel finally people are paying attention to.
Here what you're talking about is a person's own immune cells which can be either engineered or redirected to control the tumors much better. And that is something which is very exciting. And even a small number of cells have got the power to go and find the tumor and kill it.
Immune checkpoint blockade has been an exciting game changer in cancer immunotherapy. As you can imagine, you've probably seen headline news on how exciting it is. And so I'll be more specific. PD-1 blockade therapies as well as CTLA-4 blockade therapies have unleashed the T cells in the tumor to be reactive again. So the tumor actually upregulates CTLA-4 as well as PD-L1, which inhibits that, and it binds to PD-1 and CTLA-4 on the T cell. So when we use an antibody against that, it blocks that interaction and allows the T cell, which is otherwise dysfunctional because of the tumor, to be very effective. So effective that it's treated patients with melanoma, lung cancer, kidney cancer, and it's been used in a lot of solid malignancies now. It's given curative as well as durable responses to patients that would otherwise succumb to their disease.
Two checkpoints that now have been in clinical development. One is so-called CTLA-4, and the other one is called PD-1. Blockade of CTLA-4 and PD-1 have been shown to be effective broadly against many types of cancers, including melanoma, lung cancer, bladder cancer, non-Hodgkin's lymphoma, triple-negative breast cancer.
Adoptive T-cell transfer therapy is basically taking T cells from a patient, normally from the tumor of a patient, and then inside that tumor there are T cells that are actually very tumor reactive. But again, they're suppressed by the tumor. So if we enrich those T cells from the patient, we can activate them, expand them to large numbers, and put them back into the patient. And what we've seen is it can also elicit long-lasting anti-tumor responses. And what's really exciting is that at the Medical University of South Carolina, we have a clean cell facility so that we can actually do this very exciting technology and actually put it into patients.
So the clean cell facility that we have in here is one in, I think, 12 or 14 of those kind are there in the US. And this is a great facility because it can cater to the needs of immunotherapy by using the cellular therapies for T cells, dendritic cells, mesenchymal stem cells, a bunch of those things which can be used for immunotherapy can be made into the patient grade material can be made and can be transferred into the cells. So we have qualified technicians for that. We have a QA/QC person who kind of looks into with every detail so that the SOPs are implemented to the T, and then those can be used for the clinicians to go and use this facility to produce the cells which can be used for the immunotherapy. We are already working with a couple of on protocols to standardize them, and what it does is, like, if there's some kind of bench-side research which has got some promise, we validate by working with the PI to see that those can be transferred to the larger scale, do the validation runs with the GMP grade reagents, which are FDA approved, that those can be used for the patients. And then once we get those through, we have to file for PI has to file for the IND, and we help and assist them in doing so, and making the protocols through, getting the SOPs through, so that this can be implemented back and used for the patients to the bedside.
Combining adoptive T-cell transfer therapy with checkpoint modulators is going to be the next step. In fact, the National Institutes of Health, as well as places like the University of Pennsylvania, are already gearing up to do this, and that's something that we are very passionate about doing here as well. And really, it's a very timely question. So, and it makes sense to actually put the T-cells in that are tumor-specific into the patient. We know that we can actually harness a specific immune response against that patient. And inevitably, unfortunately, even the cells we infuse can become quite tired or suppressed by the tumor. And so we're getting quite clever, because now we're going to come in with checkpoint blockade. So we know the cells that are effective, we put them in there, and we can change the biology of those cells, and on top of that, keep them going. So it's, in my opinion, like a power punch.
MUSC is in a very strategic, unique position to really move the field and contribute to the field. About 6 years ago, MUSC, Hollins Cancer Center, attained the National Cancer Institute designated cancer center status. So cancer immunology program is one of the original programs to achieve such a designation, which is really prestigious. At this point, the cancer immunology program is very active. We have about 20 investigators working day and night on understanding the immune system, particularly in dealing with cancer. We have funding from National Cancer Institute, we have funding from Department of Defense, as well as funding from private foundations. We're working in a number of areas, both in understanding the fundamental aspect of immune response against cancer, but also on how to translate our knowledge to novel clinical trials.
There's a lot of creative, clever investigators. That's the other thing that's great about MUSC. There's a huge critical mass of T-cell immunologists and translational physicians that are teaming up in a really amazing way to make adoptive immunotherapy better than it already is.
So immunotherapy definitely has a lot of promise, and MUSC has all the potential in terms of expertise of investigators and the facilities that are needed to generate these cells optimally in a manner which can be put into the patients. And then there's a very dedicated set of clinicians, who are ready to, interface with the scientists and take it to that level, because it takes a lot of effort. It's a whole team.
My message to physicians is to be really interactive with basic scientists, other physicians, and I suspect that's already happening, but just be excited, because I think they're going to see they already are seeing remarkable responses in the patients. And if we understand how that's actually working, we can make it even better, because even though it's exciting and there's a lot of good news out there, there are patients that aren't responding to these therapies. Why? And that's where we need to go, is figuring out that why, so that we open the door to treat everybody.
As a physician, as well as a scientist, our obligation is to deliver medicine, but also to invent medicine. We are very excited to have the opportunity to work with everyone to not only deliver some of the cutting-edge treatment to our patients, but also collectively to advance the field by conducting, for example, clinical trials, by answering some of the important questions that are yet to be answered.